The jugal region, or nila, is a region of the wing that is usually a small membranous area proximal to the base of the vanus strengthened by a few small, irregular vein-like thickenings, but when well developed it is a distinct section of the wing and may contain one or two jugal veins. When the jugal area of the forewing is developed as a free lobe, it projects beneath the humeral angle of the hindwing and thus serves to yoke the two wings together. In the Jugatae group of Lepidoptera it bears a long finger-like lobe. The jugal region was termed the nila, new wing, because it is evidently a secondary and recently developed part of the wing. 15. The axillary region is region containing the axillary sclerites has in general the form of a scalene triangle. The base of the triangle AB is the hinge of the wing with the body. The apex C is the distal end of the third axillary sclerite. The longer side is anterior to the apex. The point D on the anterior side of the triangle marks the articulation of the radial vein with the second axillary sclerite. The line between D and C is the plica basilis BF, or fold of the wing at the base of the mediocubital field. 15. The termen is the outer margin of the wing, between apex and hind or anal angle. 17. 18. At the posterior angle of the wing base in some Diptera there is a pair of membranous lobes, squamae, or calypters, known as the alula. The alula is well developed in the housefly. The outer squama C arises from the wing base behind the third axillary sclerite, 3-ax, and evidently represents the jugal lobe of other insects. A. D. The larger inner squama D arises from the posterior scutellar margin of the tergum of the wing-bearing segment and forms a protective, hood-like canopy over the halter. In the flexed wing the outer squama of the alula is turned upside down above the inner squama, the latter not being affected by the movement of the wing. In many Diptera a deep incision of the anal area of the wing membrane behind the single vanal vein sets off a proximal alar lobe distal to the outer squama of the alula. 15. Joints. The various movements of the wings, especially in insects that flex the wings horizontally over the back when at rest, demand a more complicated articular structure at the wing base than a mere hinge of the wing with the body. Each wing is attached to the body by a membranous basal area, but the articular membrane contains a number of small articular sclerites, collectively known as the pterolia. The pterolia include an anterior humeral plate at the base of the costal vein, a group of axillaries associated with the subcostal, radial, and vanal veins, and two less definite median plates M at the base of the mediocubital area. The axillaries are specifically developed only in the wing flexing insects, where they constitute the flexor mechanism of the wing operated by the flexor muscle arising on the pleuron. Characteristic of the wing base is also a small lobe on the anterior margin of the articular area proximal to the humeral plate, which, in the forewing of some insects, is developed into a large, flat, scale-like flap, the tegula, overlapping the base of the wing. Posteriorly the articular membrane often forms an ample lobe between the wing and the body, and its margin is generally thickened and corrugated, giving the appearance of a ligament, the so-called axillary cord, continuous mesially with the posterior marginal scutellar fold of the tergal plate bearing the wing. 15. The articular sclerites, or pterolia, of the wing base of the wing flexing insects and their relations to the body and the wing veins, shown diagrammatically, are as follows. Humeral plates. First axillary. Second axillary. Third axillary. Fourth axillary. Median plates M. The humeral plate is usually a small sclerite on the anterior margin of the wing base, movable and articulated with the base of the costal vein. Odonata have their humeral plate greatly enlarged, 15, with two muscles arising from the episternum inserted into the humeral plates and two from the edge of the epimeron inserted into the axillary plate. 11. The first axillary sclerite, lax, is the anterior hinge plate of the wing base. Its anterior part is supported on the anterior nodal wing process of the tergum ANP, its posterior part articulates with the tergal margin. The anterior end of the sclerite is generally produced as a slender arm, the apex of which E is always associated with the base of the subcostal vein SC, though it is not united with the latter. The body of the sclerite articulates laterally with the second axillary. The second axillary sclerite 2-ax is more variable in form than the first axillary, but its mechanical relations are no less definite. It is obliquely hinged to the outer margin of the body of the first axillary, and the radial vein R is always flexibly attached to its anterior end D. The second axillary presents both a dorsal and a ventral sclerotization in the wing base. The second axillary presents both a dorsal and a ventral sclerotization in the wing base. Its ventral surface rests The second axillary presents both a dorsal and a ventral sclerotization in the wing base. Its ventral surface rests upon the fulcral wing process of the pleuron. The second axillary, therefore, is the pivotal sclerite of the wing base, and it specifically manipulates the radial vein. 15. The third axillary sclerite 3-ax lies in the posterior part of the articular region of the wing. Its form is highly variable and often irregular, but the third axillary is the scleroid on which is inserted the flexor muscle of the wing D. Mesially it articulates anteriorly F with the posterior end of the second axillary, and posteriorly B with the posterior wing process of the tergum P and P, or with a small fourth axillary when the latter is present. Distally the third axillary is prolonged in a process which is always associated with the bases of the group of veins in the anal region of the wing, here termed the vanal veins V. The third axillary, therefore, is usually the posterior hinge plate of the wing base and is the active scleroid of the flexor mechanism, which directly manipulates the vanal veins. The contraction of the flexor muscle T revolves the third axillary on its mesial articulations B, F and thereby lifts its distal arm. The contraction of the flexor muscle T revolves the third axillary on its mesial articulations B, F and thereby lifts its distal arm. This movement produces the flexion of the wing. The fourth axillary scleroid is not a constant element of the wing base. When present it is usually a small plate intervening between the third axillary and the posterior nodal wing process and is probably a detached piece of the latter. 15. The median plates M, are also scleroids that are not so definitely differentiated as specific plates as are the three principal axillaries, but nevertheless they are important elements of the flexor apparatus. They lie in the median area of the wing base distal to the second and third axillaries and are separated from each other by an oblique line, BF, which forms a prominent convex fold during flexion of the wing. The proximal plate M is usually attached to the distal arm of the third axillary and perhaps should be regarded as a part of the latter. The distal plate M is less constantly present as a distinct scleroid and may be represented by a general sclerotization of the base of the mediocubital field of the wing. When the veins of this region are distinct at their bases, they are associated with the outer median plate. 15. Muscles. The diamond-shaped alary muscles green of the mosquito Anopheles gambii and their structural relationship to the tube-like heart also in green. Red depicts pericardial cells, blue cell nuclei. The muscles that control flight in insects can take up to 10% to 30% of the total body mass. The muscles that control flight vary with the two types of flight found in insects, indirect and direct. Insects that use first, indirect, have the muscles attached to the tergum instead of the wings, as the name suggests. As the muscles contract, the thoracic box becomes distorted, transferring the energy to the wing. There are two bundles of muscles, those that span parallel to the tergum, the dorsolongitudinals, and those that are attached to the tegum and extend to the sternum, the dorsoventrals. 19. Indirect muscle. The connection is directly from the pleuron thoracic wall to individual scleroids located at the base of the wing. The suballar and basilar muscles have ligament attachments to the suballar and basilar scleroids. Here resilin, a highly elastic material, forms the ligaments connecting flight muscles to the wing apparatus. In more derived orders of insects, such as Diptera flies and Hymenoptera wasp, the indirect muscles occupy the greatest volume of the pterothorax and function as the primary source of power for the wing stroke. Contraction of the dorsolongitudinal muscles causes the severe arching of the notum which depresses the wing, while contraction of the dorsoventral muscles causes opposite motion of notum. The most primitive extant flying insects, Ephemeroptera mayflies and Odonata dragonflies, use direct muscles that are responsible for developing the needed power for the up and down strokes. 19-20. Insect wing muscle is a strictly aerobic tissue. Per unit protein it consumes fuel and oxygen at rates taking place in a very concentrated and highly organized tissue so that the steady-state rates per unit volume represent an absolute record in biology. The fuel and oxygen-rich blood is carried to the muscles through diffusion occurring in large amounts, in order to maintain the high level of energy used during flight. Many wing muscles are large and may be as large as 10 mm in length and 2 mm in width. Moreover, in some Diptera the fibers are of giant dimensions. For instance, in the very active Rudelia, the cross-section is 1,800 micrometers long and more than 500 micrometers wide. The transport of fuel and oxygen from the surroundings to the sites of consumption and the reverse transport of carbon dioxide therefore represent a challenge to the biologist both in relation to transport in the liquid phase and in the intricate system of air tubes, i.e. in the tracheal system. 21. Sensors. Several types of sensory neurons are found on insect wings. Gustatory bristles, 22 mechanosensory bristles, 23 campaniform sensilla, 24 and chordotonal organs. 25 These sensors provide the nervous system with both external and internal proprioceptive feedback necessary for effective flight. 26 and grooming. 27. Coupling, folding, and other features. In many insect species, the forewing and hindwing can be coupled together, which improves the aerodynamic efficiency of flight by joining the forewing and hindwing into one bigger wing. The most common coupling mechanism, e.g., Hymenoptera and Trichoptera, is a row of small hooks on the forward margin of the hindwing, or homily, which lock onto the forewing, keeping them held together, hamulate coupling. In some other insect species, e.g., Mecoptera, Lepidoptera, and some Trichoptera, the jugal lobe of the forewing covers a portion of the hindwing, jugal coupling, or the margins of the forewing and hindwing overlap broadly, amplexiform coupling, or the hindwing bristles, or frenulum, hook under the retaining structure or retinaculum on the forewing. 1443. When at rest, the wings are held over the back in most insects, which may involve longitudinal folding of the wing membrane and sometimes also transverse folding. Folding may sometimes occur along the flexion lines. Though fold lines may be transverse, as in the hind wings of beetles and earwigs, they are normally radial to the base of the wing, allowing adjacent sections of a wing to be folded over or under each other. The commonest fold line is the jugal fold, situated just behind the third anal vein, 12, although most Neoptera have a jugal fold just behind vein 3a on the forewings. It is sometimes also present on the hind wings. Where the anal area of the hindwing is large, as in Orthoptera and Blatodia, the whole of this part may be folded under the anterior part of the wing along a vanal fold a little posterior to the claval furrow. In addition, in Orthoptera and Blatodia, the anal area is folded like a fan along the veins, the anal veins being convex, at the crests of the folds, and the accessory veins concave. Whereas the claval furrow and jugal fold are probably homologous in different species, the vanal fold varies in position in different taxa. Folding is produced by a muscle arising on the pleuron and inserted into the third axillary scleroid in such a way that, when it contracts, the scleroid pivots about its points of articulation with the posterior nodal process and the second axillary scleroid. 11. As a result, the distal arm of the third axillary scleroid rotates upwards and inwards, so that finally its position is completely reversed. The anal veins are articulated with this scleroid in such a way that when it moves they are carried with it and become flexed over the back of the insect. Activity of the same muscle in flight affects the power output of the wing, and so it is also important in flight control. In orthopteroid insects, the elasticity of the cuticle causes the vanal area of the wing to fold along the veins. Consequently, energy is expended in unfolding this region when the wings are moved to the flight position. In general, wing extension probably results from the contraction of muscles attached to the basilar scleroid or, in some insects, to the suballar scleroid. 11. Flight. Australian emperor in flight, it uses the direct flight mechanism. Flight mechanisms. Two groups of relatively large insects, the Ephemeroptera mayflies and the Odonata dragonflies and damselflies, have the flight muscles attached directly to their wings. The wings can beat no faster than the rate at which nerves can send impulses to command the muscles to beat. 28. All other living winged insects fly using a different mechanism, involving indirect flight muscles which cause the thorax to vibrate. The wings can beat faster than the rate at which the muscles receive nerve impulses. This mechanism evolved once, and is the defining feature, synapomorphy, for the infraclass Neoptera. 28. Aerodynamics. There are two basic aerodynamic models of insect flight. Most insects use a method that creates a spiraling leading edge vortex. 29-30. Some very small insects use the fling and clap or Weiss-Faux mechanism, in which the wings clap together above the insect's body and then fling apart. As they fling open, the air gets sucked in and creates a vortex over each wing. This bound vortex then moves across the wing and, in the clap, acts as the starting vortex for the other wing. Circulation and lift are increased, at the price of wear and tear on the wings. 29-30. Many insects can hover by beating their wings rapidly, requiring sideways stabilization as well as lift. 31. A few insects use gliding flight, without the use of thrust. Evolution. Sometime in the Carboniferous period, some 350 million years ago, when there were only two major land masses, insects began flying. How and why insect wings developed, however, is not well understood, largely due to the scarcity of appropriate fossils from the period of their development in the lower Carboniferous. Three main theories on the origins of insect flight are that wings developed from pteranodal lobes, extensions of the thoracic terga, that they are modifications of movable abdominal gills as found on aquatic naiads of mayflies, or that they developed from thoracic protrusions used as radiators. 32. Fossils. Holotype wing of the extinct Cymbroflebia brooksii. 49.5 million years old, Boot Hill, Klondike Mountain Formation, Washington, USA. Fossils from the Devonian 400 million years ago are all wingless, but by the Carboniferous 320 million years ago, more than 10 different genera of insects had fully functional wings. There is little preservation of transitional forms between the two periods. The earliest winged insects are from this time period Pterygota, including the Blatoptera, Colonerodia, primitive stem group Ephemeropterans, Orthoptera and Paleodictyopteroidea. Very early Blatopterans during the Carboniferous had a very large discoid pronotum and coriaceous forewings with a distinct cup vein, an unbranched wing vein, lying near the claval fold and reaching the wing posterior margin. 33. 399. Even though the oldest possible insect fossil is the Devonian Rhyniagnatha hursti, estimated at 396 to 407 million years old, it possessed decondylate mandibles, a feature associated with winged insects. 34. Although it is later considered as possible myriapod. 35. During the Permian, the dragonflies Odonata were the dominant aerial predator and probably dominated terrestrial insect predation as well. True Odonata appeared in the Permian. 36, 37, and all are amphibious. Their prototypes are the oldest winged fossils. 38 go back to the Devonian, and are different from other wings in every way. 39 Their prototypes may have had the beginnings of many modern attributes even by late Carboniferous, and it is possible that they even captured small vertebrates, for some species had a wingspan of 71 centimeters. 37 The earliest beetle-like species during the Permian had pointed, leather-like forewings with cells and pits. Hemiptera, or true bugs, had appeared in the form of Arctonischidina and Paranidia, having forewings with unusual venation, possibly diverging from Blatoptera. 33 186 A single large wing from a species of Diptera in the Triassic, 10 mm instead of usual 2 to 6 mm, was found in Australia. M.T. Crosby. This family Tiliardipteridae, despite the numerous tipuloid features, should be included in Psychotomorpha sensu Hennig on account of loss of the convex distal 1A reaching wing margin and formation of the anal loop. 40. Hypotheses. Diagram of different theories. A hypothetical wingless ancestor. B. Piranodal theory. Hypothetical insect with wings from the back. Notum. C. Hypothetical insect with wings from the pleroma. D. Epicoxal theory. Hypothetical insect with wings from annex of the legs. 1. Notum, back. 2. Pleroma. 3. Excite, outer attachments of the legs. Piranodal hypothesis. This hypothesis suggests that the insect's wings developed from piranodal lobes, a preadaptation found in insect fossils that is believed to have assisted stabilization while hopping or falling. In favor of this hypothesis is the tendency of most insects, when startled while climbing on branches, to escape by dropping to the ground. Such lobes would have served as parachutes and enable the insect to land more softly. The theory suggests that these lobes gradually grew larger and in a later stage developed a joint with the thorax. Even later would appear the muscles to move these crude wings. This model implies a progressive increase in the effectiveness of the wings, starting with parachuting, then gliding and finally active flight. Still, lack of substantial fossil evidence of the development of the wing joints and muscles poses a major difficulty to the theory, as does the seemingly spontaneous development of articulation and venation. 32. Epicoxal hypothesis. This theory, first proposed in 1870 by Carl Gegenbauer, suggested that a possible origin for insect wings might have been the movable abdominal gills found in many aquatic insects, such as on naiads of mayflies. 41. According to this theory, these tracheal gills, which started their way as exits of the respiratory system and over time were modified into locomotive purposes, eventually developed into wings. The tracheal gills are equipped with little winglets that perpetually vibrate and have their own tiny straight muscles. 32. Indite-excite hypothesis. This hypothesis stems from the adaptation of indites and excites, appendages on the respective inner and outer aspects of the primitive arthropod limb. It was advanced by Truman, 42, based on a study by Goldschmidt in 1945 on Drosophila melanogaster, in which a pod variation displayed a mutation transforming normal wings to what was interpreted as a triple-jointed leg arrangement with some additional appendages but lacking the tarsus, where the wing's costal surface normally would be. This mutation was reinterpreted as strong evidence for a dorsal excite and indite fusion, rather than a leg, with the appendages fitting in much better with this hypothesis. The innervation, articulation and musculature required for the evolution of wings are already present in podomers. 32. Piranota plus leg gene recruitment hypothesis, also known as the dual origin hypothesis. The fossil larva of Coxoplectoptera provided important new clues to the disputed question of the evolutionary origin of insect wings. Before the larva fossil discovery, the piranodal hypothesis and the leg excite hypothesis have been considered as incompatible alternative explanations, which have both been supported by a set of evidences from the fossil record, comparative morphology, developmental biology and genetics. The expression of leg genes in the ontogeny of the insect wing has been universally considered as conclusive evidence in favor of the leg excite hypothesis, which proposes that insect wings are derived from mobile leg appendages, excites. However, the larvae of Coxoplectoptera show that the abdominal gills of mayflies and their ancestors, which are generally considered as corresponding structures to insect wings, are articulated within the dorsal tergite plates. This cannot be seen in modern mayfly larvae, because their abdominal tergites and sternites are fused to rings, without any traces left even in embryonic development. If larval gills and wings are corresponding, serial homologous structures and thus share the same evolutionary origin, the new results from Coxoplectoptera demonstrate that also wings are of tergal origin, as proposed by the classical paranodal hypothesis. Stanishek, Bechley and Godunko 2011 32-43 therefore suggested a new hypothesis that could reconcile the apparently conflicting evidence from paleontology and developmental genetics. Wings first originated as stiff outgrowths of tergal plates paranoda and only later in evolution became mobile, articulated appendages through secondary recruiting of leg genes. 32. More recent fossil analysis of Paleozoic nymph wing pads provides additional support for the fusion of the paranoda elements and arthropodon leg genes. 44. Evolution of the ways the wings at rest to the body to create. Wings do not fold back. Recent Archaeoptera spread laterally, large bubbles over the back against one another. Damselflies, mayflies. Folding. Neoptera wings not foldable, e.g., stoneflies. Folding. Fanfold, e.g., front wings of wasps. Crossfold, such as the rear wing of the beetle. Subjects folding, such as the rear wing of the earwigs. Suggestions have been made that wings may have evolved initially for sailing on the surface of water as seen in some stoneflies. 45. An alternative idea is that it derives from directed aerial gliding descent, a preflight phenomena found in some apterygote, a wingless sister taxa to the winged insects. 46. The earliest flyers were similar to dragonflies with two sets of wings, direct flight muscles, and no ability to fold their wings over their abdomens. Most insects today, which evolved from those first flyers, have simplified to either one pair of wings or two pairs functioning as a single pair and using a system of indirect flight muscles. 32. Natural selection has played an enormous role in refining the wings, control and sensory systems, and anything else that affects aerodynamics or kinematics. One noteworthy trait is wing twist. Most insect wings are twisted, as are helicopter blades, with a higher angle of attack at the base. The twist generally is between 10 and 20 degrees. In addition to this twist, the wing surfaces are not necessarily flat or featureless. Most larger insects have wing membranes distorted and angled between the veins in such a way that the cross-section of the wings approximates an airfoil. Thus, the wing's basic shape already is capable of generating a small amount of lift at zero angle of attack. Most insects control their wings by adjusting tilt, stiffness, and flapping frequency of the wings with tiny muscles in the thorax below. Some insects evolved other wing features that are not advantageous for flight, but play a role in something else, such as mating or protection. 32. Some insects occupying the biological niches that they do need to be incredibly maneuverable. They must find their food in tight spaces and be capable of escaping larger predators, or they may themselves be predators and need to capture prey. Their maneuverability, from an aerodynamic viewpoint, is provided by high lift and thrust forces. Typical insect flyers can attain lift forces up to 3 times their weight and horizontal thrust forces up to 5 times their weight. There are two substantially different insect flight mechanisms, and each has its own advantages and disadvantages. Just because odonats have a more primitive flight mechanism does not mean they are less able flyers. They are, in certain ways, more agile than anything that has evolved afterward. 32. Morphogenesis. While the development of wings in insects is clearly defined in those who are members of Endopterygota, which undergo complete metamorphosis, in these species, the wing develops while in the pupal stage of the insect's life cycle. However, insects that undergo incomplete metamorphosis do not have a pupal stage, therefore they must have a different wing morphogenesis. Insects such as those that are hemimetabolic have wings that start out as buds, which are found underneath the exoskeleton, and do not become exposed until the last instar of the nymph. 47. The first indication of the wing buds is of a thickening of the hypodermis, which can be observed in insect species as early the embryo, and in the earliest stages of the life cycle. During the development of morphological features while in the embryo, or embryogenesis, a cluster of cells grow underneath the ectoderm which later in development, after the lateral ectoderm has grown dorsally to form wind imaginal disc. An example of wing bud development in the larva can be seen in those of white butterflies Pieris. In the second instar the histoblast become more prominent, which now form a pocket-like structure. As of the third and fourth instars, the histoblast become more elongated. This greatly extended and evaginated, or protruding, part is what becomes the wing. By the close of the last instar, or fifth, the wing is pushed out of the wing pocket, although continues to lie under the old larval cuticle while in its prepupal stage. It is not until the butterfly is in its pupal stage that the wing bud becomes exposed, and shortly after eclosion, the wing begins to expand and form its definitive shape. 47. The development of tracheation of the wings begin before the wing histoblast form, as it is important to note that they develop near a large trachea. During the fourth instar, cells from the epithelium of this trachea become greatly enlarged extend into the cavity of the wing bud, with each cell having developed a closely coiled tracheole. Each trachecole is of unicellular origin, and is at first intracellular in position, while tracheae are of multicellular origin and the lumen of each is intercellular in position. The development of tracheoles, each coiled within a single cell of the epithelium of a trachea, and the subsequent opening of communication between the tracheoles and the lumen of the trachea, and the uncoiling and stretching out of the tracheoles, so that they reach all parts of the wing. 47. In the earlier stages of its development, the wing bud is not provided with special organs of respiration such as tracheation, as it resembles in this respect the other portions of the hypodermis of which it is still a part. The histoblast is developed near a large trachea, a cross-section of which is shown in, which represents sections of these parts of the first, second, third and fourth instars respectively. At the same time the tracheoles uncoil, and extend in bundles in the forming vein cavities of the wing bud. At the molt that marks the beginning of the pupal stadium stage, they become functional. At the same time, the larval tracheoles degenerate, their function having been replaced by the wing tracheae. 47. Nomenclature. Most of the nomenclature of insect orders is based on the ancient Greek word for wing, pteron, as the suffix ptera. Scientific name linguistic group. Adaptations. Variation. Insect wings are fundamental in identifying and classifying species as there is no other set of structures in studying insects more significant. Each order and insect family has distinctive wing shapes and features. In many cases, even species may be distinguished from each other by differences of color and pattern. For example, just by position one can identify species, albeit to a much lesser extent. Though most insects fold their wings when at rest, dragonflies and some damselflies rest with their wings spread out horizontally, while groups such as the caddisflies, stoneflies, alderflies, and lacewings hold their wings sloped roof-like over their backs. A few moths wrap their wings around their bodies, while many flies and most butterflies close their wings together straight upward over the back. 48. Many times the shape of the wings correlates with the type of insect flight. The best flying insects tend to have long, slender wings. In many species of Sphingidae, sphinx moths, the forewings are large and sharply pointed, forming with the small hind wings a triangle that is suggestive of the wings of fast, modern airplanes. Another, possibly more important correlation, is that of the size and power of the muscles to the speed and power of flight. In the powerfully flying insects, the wings are most adapted for the stresses and aerodynamics of flight. The veins are thicker, stronger, and closer together toward the front edge, or leading edge, and thinner yet flexible toward the rear edge, or trailing edge. This makes the insect wing an excellently constructed airfoil, capable of exerting both propulsion and lift while minimizing drag. 48. Variation of the wing beat may also occur, not just amongst different species, but even among individuals at different times. In general, the frequency is dependent upon the ratio between the power of the wing muscles and the resistance of the load. Large-winged, light-bodied butterflies may have a wing beat frequency of 4 to 20 per second, whereas small-winged, heavy-bodied flies and bees beat their wings more than 100 times a second, and mosquitoes can beat up to 988-1046 times a second. The same goes for flight. Though it is generally difficult to estimate the speed of insects in flight, most insects can probably fly faster in nature than they do in controlled experiments. 48. Coleoptera. In species of Coleoptera beetles, the only functional wings are the hind wings. The hind wings are longer than the elytra, folded longitudinally and transversely under the elytra. The wing is rotated forwards on its base into flight position. This action spread the wing and unfolded longitudinally and transversely. There is the spring mechanism in the wing structure, sometimes with the help of abdomen movement, to keep the wing in folded position. The beetle wing venation is reduced and modified due to the folding structure, which include 49. Cross-folding in the wings of beetles. The hindwing, spread, by folding lines, it is divided into 5 fields that are completed each to the rear. The same wing, half-folded, the 2 joints of the cross-folding form an obtuse angle. The right is already in the wings folded in 3 layers. With greater resolution, the 3rd arch of the wing margin in the 1st and 2nd is visible. To the left of the 5th arch appears in the 4th. The same wing, folded completely, the 5 fields are aligned, the elytra have been removed. Costa C, subcosta posterior, SCP, at the leading wing marginal, fused for most of the length. Radius anterior, RA, divided into 2 branches beyond the middle of the wing. Radius posterior, RP, basal connection is lost. Media posterior, MP, branches, long and strong vein. Cubitus anterior, CUA. Anal veins, AA, AP, veins behind the cubitus, separated by anal fold. In most species of beetles, the front pair of wings are modified and sclerotized, hardened, to form elytra, and they protect the delicate hind wings which are folded beneath. 33. The elytra are connected to the pterothorax, being called as such because it is where the wings are connected, pteron meaning wing, in Greek. The elytra are not used for flight, but tend to cover the hind part of the body and protect the second pair of wings, alae. The elytra must be raised in order to move the hind flight wings. A beetle's flight wings are crossed with veins and are folded after landing, often along these veins, and are stored below the elytra. In some beetles, the ability to fly has been lost. These include some ground beetles, family Carabidae, and some, true weevils, family Curculionidae, but also some desert and cave-dwelling species of other families. Many of these species have the two elytra fused together, forming a solid shield over the abdomen. In a few families, both the ability to fly and the elytra have been lost, with the best known example being the glowworms of the family Phengodidae, in which the females are larviform throughout their lives. 1333. Lepidoptera. Transition of scales color on a butterfly wing, 30x magnification. The two pairs of wings are found on the middle and third segment, or mesothorax and metathorax respectively. In the more recent genera, the wings of the second segment are much more pronounced, however some more primitive forms have similarly sized wings of both segments. The wings are covered in scales arranged like shingles, forming the extraordinary variety seen in color. The mesothorax is evolved to have more powerful muscles to propel moth or butterfly through the air, with the wing of said segment having a stronger vein structure. 33. 560. The largest superfamily, Noctuidae, has the wings modified to act as tympanal or hearing organs. 50. Modifications in the wing's venation include 49. Costa C, not found in butterflies. Subcosta SC plus radius 1, SC plus R1, at the leading wing marginal, fused or very close for most of the length, in hindwing fused and well developed in the humeral area. Subcosta never branches in butterfly. Radius R2 R5, radius divides into branches beyond the middle of the wing up to 5 branches in Populionidae. On forewing, the last R is stalked in all butterflies except Hesperiidae is separated. Radius sector Rs in hindwing. Media M1 M3, the basal section has been lost. Cubitus anterior CUA1 CUA2, cup section has been lost. Anal veins A, 1A plus 2A, 3A, either one vein A, or two veins 1A plus 2A, 3A. Humeral vein, the hindwing of most butterflies has a humeral vein, except Lycaenidae there is the enlargement of the humeral area of the hindwing which is overlapped with the forewing. The humeral vein strengthened the hindwing overlapped area so that the two wings coupling better. The wings, head parts of thorax and abdomen of Lepidoptera are covered with minute scales, from which feature the order Lepidoptera, derives its names, the word, lepteron, in ancient Greek meaning scale. Most scales are lamellar, or blade-like and attached with a pedicel, while other forms may be hair-like or specialized as secondary sexual characteristics. 51. The lumen or surface of the lamella, has a complex structure. It gives color either due to the pigmentary colors contained within or due to its three-dimensional structure. 52. Scales provide a number of functions, which include insulation, thermoregulation, aiding gliding flight, amongst others, the most important of which is the large diversity of vivid or indistinct patterns they provide which help the organism protect itself by camouflage, mimicry, and to seek mates. 51. Odonata. Species of Odonata, damselflies and dragonflies both have two pairs of wings which are about equal in size and shape and are clear in color. There are 5, if the R plus M is counted as 1. Main vein stems on dragonfly and damselfly wings, and wing veins are fused at their bases and the wings cannot be folded over the body at rest, which also include 49. Dragonfly. Damselfly. Costa C at the leading edge of the wing, strong and marginal, extends to the apex of the wing. Subcosta SC, second longitudinal vein, it is unbranched, joins C at nodus. Radius and media R plus M, third and fourth longitudinal vein, the strongest vein on the wing, with branches, R1 R4, reach the wing margin, the media anterior MA or also reach the wing margin. IR2 and IR3 are intercalary veins behind R2 and R3 respectively. Cubitus CU, fifth longitudinal vein, cubitus posterior cup, is unbranched and reach the wing margin. Anal veins A1, unbranched veins behind the cubitus. A nodus is formed where the second main vein meets the leading edge of the wing. The black pterostigma is carried near the wing tip. The main veins and the crossveins form the wing venation pattern. The venation patterns are different in different species. There may be very numerous crossveins or rather few. The Australian flatwing damselfly's wings are one of the few veins patterns. The venation pattern is useful for species identification. 49. Almost all Anisoptera settle with the wings held out sideways or slightly downward, however most Zygoptera settle with the wings held together, dorsal surfaces opposed. The thorax of Zygoptera is so oblique that when held in this way the wings fit neatly along the top of the abdomen. They do not appear to be held straight up as in butterflies or mayflies. In a few Zygopteran families the wings are held horizontally at rest, and in one Anisopteran genus e.g. Corduliidae, the wings are held in the typical damselfly resting position. Adult species possess 2 pairs of equal or subequal wings. There appear to be only 5 main vein stems. A nodus is formed where the second main vein, subcosta, meets the leading edge of the wing. In most families a conspicuous pterostigma is carried near the wingtip. Identification as Odonata can be based on the venation. The only likely confusion is with some lacewings, order Neuroptera, which have many crossveins in the wings. Until the early years of the 20th century Odonata were often regarded as being related to lacewings and were given the ordinal name Paraneroptera, but any resemblance between these two orders is entirely superficial. In Anisoptera the hindwing is broader than the forewing and in both wings a crossvein divides the discoidal cell into a triangle and supertriangle. 53. Orthoptera. Species of Orthoptera, grasshoppers and crickets, have forewings that are tough opaque tegmina, narrow which are normally covering the hind wings and abdomen at rest. The hind wings are bored membranous and folded in fan-like manner, which include the following venation. 49 Costa C, at the leading marginal of the forewing and hindwing, unbranched. Subcosta SC, second longitudinal vein, unbranched. Radius R, third longitudinal vein, branched to Rs in forewing and hindwing. Media anterior MA, fourth longitudinal vein, branched in basal part as media posterior MP. Cubitus CU, fifth longitudinal vein, on forewing and hindwing dividing near the wing base into branched cua, an unbranched cup. Anal veins 8 veins behind the cubitus, unbranched, 2 in forewing, many in hindwing. Phasmatodia. Costa C, at the leading marginal of the hindwing, unbranched, absent in forewing. Subcosta SC, second longitudinal vein, unbranched. Radius R, third longitudinal vein, branched to Rs in hindwing, unbranched in forewing. Media anterior MA, fourth longitudinal vein, branched in basal part as media posterior MP. Cubitus CU, fifth longitudinal vein, unbranched. Anal veins 8 veins behind the cubitus, unbranched, 2 in forewing, many in hindwing, 1A7A in one group and the rest in another group. Stick insect have forewings that are tough, opaque tegmina, short and covering only the base part of the hind wings at rest. Hindwings from costa to cubitus are tough and opaque like the forewings. The large anal area are membranous and folded in fan-like manner. There are no or very few branching in stick insect wing veins. 49. Dermaptera. Unfolding of earwig wing. The front and rear wings at rest. The front wing covers most of the hindwing, with only the joint projects in the form of a quarter circle forward with a central white spot under the forewing. On the right-hand side of the forewing is open to the right blue arrow, which from this perspective appears narrower than it is with the rear wing still folded completely. The front wing is open to the left blue arrow, with the right side of the forewing removed. The hindwing is half open. With greater resolution, the multiple folding is shown, resembling a fan which is parallel to the lines B and circa. The arrow points to the E point where the fan is closed again, having been folded by 180 degrees. Other orders such as the Dermaptera, earwigs, Orthoptera, grasshoppers, crickets, Mantodia, praying mantis, and Latodia, cockroaches, have rigid leathery forewings that are not flapped while flying, sometimes called tegmen place. Tegmina, elytron, place. Elytra or pseudoelytron. 13. Hemiptera. In Hemiptera, true bugs, the forewings may be hardened, though to a lesser extent than in the beetles. For example, the anterior part of the front wings of stink bugs is hardened, while the posterior part is membranous. They are called hemelytron place. Hemelytra. They are only found in the suborder Heteroptera. The wings of the Homoptera, such as the cicada, are typically entirely membranous. Both forewings and hind wings of cicada are membranous. Most species are glass-like although some are opaque. Cicadas are not good flyers and most fly only a few seconds. When flying, forewing and hindwing are hooked together by a grooved coupling along the hindwing costa and forewing margin. Most species have a basic venation as shown in the following picture. 49. Costa C, at the leading wing marginal, in forewing extends to the node and lies close to SC plus R. Subcosta plus radius SC plus R, in forewing SC and R fuse together to the node. Radial sector Rs arises near the node and unbranches. Radius anterior RA. Radius posterior RP. Media M branches to M1 to M4. Cubitus anterior cua branches to cua 1 and cua 2. Cubitus posterior cup unbranches. Anal veins 8 veins behind the cubitus, 1A and 2A fused in the forewing, cup and 2A are folded. Also notice there are the ambient veins and peripheral membranes on the margin of both wings. Diptera. In the Diptera true flies, there is only one pair of functional wings, with the posterior pair of wings are reduced to halteres, which help the fly to sense its orientation and movement, as well as to improve balance by acting similar to gyroscopes. In Calyptridae, the very hindmost portion of the wings are modified into somewhat thickened flaps called calypters which cover the halteres. 49. Costa C, not found in Diptera. Subcosta SC became the leading wing vein, unbranched. Radius R branched to R1 R5. Media M branched to M1 M4. Cubitus anterior Kua unbranched, cup is reduced in Diptera. Some species Kua and 1A are separated, some species meets when reaching the wing margin, some species fused. Anal veins A, only two anal veins 1A and 2A are present, 2A is not distinctive in some species. Discal cell DC, well defined in most species. Blatodia. Species of Blatodia cockroaches have a forewing, or also known as tegmen, that is more or less sclerotized. It is used in flight as well as a form of protection of the membranous hind wings. The veins of hindwing are about the same as front wing but with large anal lobe folded at rest between cup and 1A. The anal lobe usually folded in a fan-like manner. 49. An example of longitudinal folding in wasps Vespidae. The main fold line of the forewing seen halfway up is a bright horizontal line. The wing part that is behind this line is turned back down. The narrow strip at the front edge of the wing is in front of the first strong wire folded forward and down. So in rest position, the outer lining forms the tough outer edge of the wing, which protects the sides of the abdomen as a shock absorber. The rear wing is covered largely by the forewing. Costa C, at the leading edge of the wing. Subcosta SC, second longitudinal vein, it is relatively short. Radius R, third longitudinal vein, with many pectinate branches. Media M, fourth longitudinal vein, reach the wing margin. Cubitus anterior Kua, fifth longitudinal vein, with dichotomous branches occupy large part of tegmen. Cubitus posterior cup is unbranched, curved and reach the wing margin. Anal veins A, veins behind the cubitus. Hymenoptera. Hymenoptera adults, including sawflies, wasps, bees, and non-worker ants, all have two pairs of membranous wings. 49. Costa C, not found in Hymenoptera. Subcosta SC, unbranched. Radius R, branched to R1 R5. Media M is unbranched, in forewing M is fused with Rs for part of its length. Cubitus Kua, unbranched, cup is absent in Hymenoptera. Anal veins A, only two anal veins 1A and 2A are present, 2A is not distinctive in some species. Wing coupling, row of hooks on the leading edge of hindwing engage the hind margin of the forewing, strongly couple the wings in flight. Line of wing folding, some species, including Vespidae, the forewing are longitudinally folded along the line of wing folding, at rest. Pterostigma, is present for some species. The forward margin of the hindwing bears a number of hooked bristles, or homily, which lock onto the forewing, keeping them held together. The smaller species may have only 2 or 3 homily on each side, but the largest wasps may have a considerable number, keeping the wings gripped together especially tightly. Hymenopteran wings have relatively few veins compared with many other insects, especially in the smaller species. 13. Other families. Termites are relatively poor flyers and are readily blown downwind in wind speeds of less than 2 km per hour, shedding their wings soon after landing at an acceptable site, where they made an attempt to form a nest in damp timber or earth. 54. Wings of most termites have 3 heavy veins along the basal part of the front edge of the forewing and the cross veins near the wing tip are angled, making trapezoidal cells. Although subterranean termite wings have just 2 major veins along the front edge of the forewing and the cross veins towards the wingtip are perpendicular to these veins, making square and rectangular cells. 55. Species of Thysanoptera, thrips, Teleidae and other flying microinsects have slender front and hind wings with long fringes of hair, called fringed wings, also referred to as teloptery. 56. While species of Trichoptera, caddisfly, have hairy wings with the front and hind wings clothed with setae. 13. Male Strepsiptera also have halteres that evolved from the forewings instead of the hind wings. This means that only their hind wings are functional at flying, as opposed to Diptera which have functional forewings and halteres for hind wings. Also the hind wings in males of Coccidae are reduced to halteres or are absent. 57. Insect flight. Insects are the only group of invertebrates that have evolved wings in flight. Insects first flew in the Carboniferous, some 300 to 350 million years ago, making them the first animals to evolve flight. Wings may have evolved from appendages on the sides of existing limbs, which already had nerves, joints, and muscles used for other purposes. These may initially have been used for sailing on water, or to slow the rate of descent when gliding. A tau emerald Hemicordalia tau dragonfly has flight muscles attached directly to its wings. Two insect groups, the dragonflies and the mayflies, have flight muscles attached directly to the wings. In other winged insects, flight muscles attach to the thorax, which make it oscillate in order to induce the wings to beat. Of these insects, some flies and some beetles achieve very high wing beat frequencies through the evolution of an asynchronous nervous system, in which the thorax oscillates faster than the rate of nerve impulses. Not all insects are capable of flight. A number of apterous insects have secondarily lost their wings through evolution, while other more basal insects like silverfish never evolved wings. In some eusocial insects like ants and termites, only the alate reproductive castes develop wings during the mating season before shedding their wings after mating, while the members of other castes are wingless their entire lives. Some very small insects make use not of steady-state aerodynamics, but of the Weis-Faux clap and fling mechanism, generating large lift forces at the expense of wear and tear on the wings. Many insects can hover, maintaining height and controlling their position. Some insects such as moths have the forewings coupled to the hind wings so these can work in unison. Mechanisms. Direct flight. Unlike other insects, the wing muscles of the Ephemeroptera, mayflies, and Odonata, dragonflies and damselflies insert directly at the wing bases, which are hinged so that a small downward movement of the wing base lifts the wing itself upward, much like rowing through the air. Dragonflies and damselflies have fore and hind wings similar in shape and size. Each operates independently, which gives a degree of fine control and mobility in terms of the abruptness with which they can change direction and speed, not seen in other flying insects. Odonats are all aerial predators, and they have always hunted other airborne insects. 1. Direct flight. Muscles attached to wings. Large insects only. The Odonata, dragonflies, and damselflies have direct flight musculature, as do mayflies. And direct flight. Other than the two orders with direct flight muscles, all other living winged insects fly using a different mechanism, involving indirect flight muscles. This mechanism evolved once and is the defining feature, synapomorphy, for the infraclass Neoptera. It corresponds, probably not coincidentally, with the appearance of a wing folding mechanism, which allows Neopteran insects to fold the wings back over the abdomen when at rest, though this ability has been lost secondarily in some groups, such as in the butterflies. 1. What all Neoptera share, however, is the way the muscles in the thorax work. These muscles, rather than attaching to the wings, attach to the thorax and deform it. Since the wings are extensions of the thoracic exoskeleton, the deformations of the thorax cause the wings to move as well. A set of longitudinal muscles along the back compresses the thorax from front to back, causing the dorsal surface of the thorax notum to bow upward, making the wings flip down. Another set of muscles from the tergum to the sternum pulls the notum downward again, causing the wings to flip upward. 1, 2. And direct flight. Muscles make thorax oscillate in most insects. The Neoptera, including butterflies and most other insects, have indirect flight musculature. Insects that beat their wings fewer than 100 times a second use synchronous muscle. Synchronous muscle is a type of muscle that contracts once for every nerve impulse. This generally produces less power and is less efficient than asynchronous muscle, which accounts for the independent evolution of asynchronous flight muscles in several separate insect clades. 3. Insects that beat their wings more rapidly, such as the bumblebee, use asynchronous muscle. This is a type of muscle that contracts more than once per nerve impulse. This is achieved by the muscle being stimulated to contract again by a release in tension in the muscle, which can happen more rapidly than through simple nerve stimulation alone. 4. This allows the frequency of wing beats to exceed the rate at which the nervous system can send impulses. The asynchronous muscle is one of the final refinements that has appeared in some of the higher Neoptera, Coleoptera, Diptera, and Hymenoptera. The overall effect is that many higher Neoptera can beat their wings much faster than insects with direct flight muscles. 1. Asynchronous muscle is, by definition, under relatively coarse control by the nervous system. To balance this evolutionary trade-off, insects that evolved in direct flight have also developed a separate neuromuscular system for fine-grained control of the wing stroke. 5. Known as direct muscles. These muscles attach directly to the sclerites that make up the wing hinge and are contracted with 1 impulses from motor neurons. 6. Recent work has begun to address the complex nonlinear muscular dynamics at the wing hinge and its effects on the wingtip path. 7. Aerodynamics. There are two basic aerodynamic models of insect flight, creating a leading edge vortex, and using clap and fling. 8-9. Leading edge vortex. Most insects use a method that creates a spiraling leading edge vortex. These flapping wings move through two basic half strokes. The downstroke starts up and back and is plunged downward and forward. Then the wing is quickly flipped over, supination, so that the leading edge is pointed backward. The upstroke then pushes the wing upward and backward. Then the wing is flipped again, pronation, and another downstroke can occur. The frequency range in insects with synchronous flight muscles typically is 5 to 200 Hz Hc. In those with asynchronous flight muscles, wing beat frequency may exceed 1000 Hz. When the insect is hovering, the two strokes take the same amount of time. A slower downstroke, however, provides thrust. 8-9. Identification of major forces is critical to understanding insect flight. The first attempts to understand flapping wings assumed a quasi-steady state. This means that the airflow over the wing at any given time was assumed to be the same as how the flow would be over a non-flapping, steady-state wing at the same angle of attack. By dividing the flapping wing into a large number of motionless positions and then analyzing each position, it would be possible to create a timeline of the instantaneous forces on the wing at every moment. The calculated lift was found to be too small by a factor of 3, so researchers realized that there must be unsteady phenomena providing aerodynamic forces. There were several developing analytical models attempting to approximate flow close to a flapping wing. Some researchers predicted force peaks at supination. With a dynamically scaled model of a fruit fly, these predicted forces later were confirmed. Others argued that the force peaks during supination and pronation are caused by an unknown rotational effect that fundamentally is different from the translational phenomena. There is some disagreement with this argument. Through computational fluid dynamics, some researchers argue that there is no rotational effect. They claim that the high forces are caused by an interaction with the wake shed by the previous stroke. 8-9. Similar to the rotational effect mentioned above, the phenomena associated with flapping wings are not completely understood or agreed upon. Because every model is an approximation, different models leave out effects that are presumed to be negligible. For example, the Wagner effect, as proposed by Herbert A. Wagner in 1925, says that circulation rises slowly to its steady state due to viscosity when an inclined wing is accelerated from rest. This phenomenon would explain a lift value that is less than what is predicted. Typically, the case has been to find sources for the added lift. It has been argued that this effect is negligible for flow with a Reynolds number that is typical of insect flight. The Reynolds number is a measure of turbulence. Flow is laminar, smooth, when the Reynolds number is low, and turbulent when it is high. 11. The Wagner effect was ignored, consciously, in at least one model. 9. One of the most important phenomena that occurs during insect flight is leading edge suction. This force is significant to the calculation of efficiency. The concept of leading edge suction first was put forth by D.G. Ellis and J.L. Stollery in 1988 to describe vortex lift on sharp-edged delta wings. 12. At high angles of attack, the flow separates over the leading edge, but reattaches before reaching the trailing edge. Within this bubble of separated flow is a vortex. Because the angle of attack is so high, a lot of momentum is transferred downward into the flow. These two features create a large amount of lift force as well as some additional drag. The important feature, however, is the lift. Because the flow has separated, yet it still provides large amounts of lift, this phenomenon is called stall delay, first noticed on aircraft propellers by H. Himmelskamp in 1945. 13. This effect was observed in flapping insect flight and it was proven to be capable of providing enough lift to account for the deficiency in the quasi-steady state models. This effect is used by canoeists in a sculling draw stroke. 8-9. All of the effects on a flapping wing may be reduced to three major sources of aerodynamic phenomena: the leading edge vortex, the steady-state aerodynamic forces on the wing, and the wing's contact with its wake from previous strokes. The size of flying insects ranges from about 20 micrograms to about 3 grams. As insect body mass increases, wing area increases and wing beat frequency decreases. For larger insects, the Reynolds number may be as high as 10,000, where flow is starting to become turbulent. For smaller insects, it may be as low as 10. This means that viscous effects are much more important to the smaller insects. 9, 14, 15. Downstroke. Upstroke. Another interesting feature of insect flight is the body tilt. As flight speed increases, the insect body tends to tilt nose down and become more horizontal. This reduces the frontal area and therefore, the body drag. Since drag also increases as forward velocity increases, the insect is making its flight more efficient as this efficiency becomes more necessary. Additionally, by changing the geometric angle of attack on the downstroke, the insect is able to keep its flight at an optimal efficiency through as many maneuvers as possible. The development of general thrust is relatively small compared with lift forces. Lift forces may be more than 3 times the insect's weight, while thrust at even the highest speeds may be as low as 20% of the weight. This force is developed primarily through the less powerful upstroke of the flapping motion. 9:16. Clap and fling. The feathery wings of a thrips are unsuitable for leading-edge vortex flight, but support clap and fling. Clap and fling is used in sea butterflies like Limacina helicina to fly through the water. Clap and fling, or the Weisfo mechanism, discovered by the Danish zoologist Turkel Weisfo, is a lift generation method utilized during small insect flight. 17. As insect sizes become less than 1 mm, viscous forces become dominant and the efficacy of lift generation from an airfoil decreases drastically. Starting from the clap position, the two wings fling apart and rotate about the trailing edge. The wings then separate and sweep horizontally until the end of the downstroke. Next, the wings pronate and utilize the leading edge during an upstroke rowing motion. As the clap motion begins, the leading edges meet and rotate together until the gap vanishes. Initially, it was thought that the wings were touching, but several incidents indicate a gap between the wings and suggest it provides an aerodynamic benefit. 18-19. Lift generation from the clap and fling mechanism occurs during several processes throughout the motion. First, the mechanism relies on a wing-wing interaction, as a single wing motion does not produce sufficient lift. 20-21-22. As the wings rotate about the trailing edge in the flinging motion, air rushes into the created gap and generates a strong leading-edge vortex, and a second one developing at the wingtips. A third, weaker, vortex develops on the trailing edge. 19. The strength of the developing vortices relies, in part, on the initial gap of the interwing separation at the start of the flinging motion. With a decreased gap interwing gap indicating a larger lift generation, at the cost of larger drag forces. The implementation of a heaving motion during fling, 23. Flexible wings, 21. And a delayed stall mechanism were found to reinforce vortex stability and attachment. 24. Finally, to compensate the overall lower lift production during low Reynolds number flight, with laminar flow, tiny insects often have a higher stroke frequency to generate wing tip velocities that are comparable to larger insects. 24. The overall largest expected drag forces occur during the dorsal fling motion, as the wings need to separate and rotate. 22. The attenuation of the large drag forces occurs through several mechanisms. Flexible wings were found to decrease the drag in flinging motion by up to 50% and further reduce the overall drag through the entire wing stroke when compared to rigid wings. 21. Bristles on the wing edges, as seen in Encarsia formosa, cause a porosity in the flow which augments and reduces the drag forces, at the cost of lower lift generation. 25. Further, the interwing separation before fling plays an important role in the overall effect of drag. As the distance increases between the wings, the overall drag decreases. 18. The clap and fling mechanism is also employed by the marine mollusk Limacina helicina, a sea butterfly. 26-27. Some insects, such as the vegetable leaf miner Liriomyza sativate, a fly, exploit a partial clap and fling, using the mechanism only on the outer part of the wing to increase lift by some 7% when hovering. 24. Clap and fling flight mechanism after Sain 2003. Fling 1. Wings rotate around trailing edge to fling apart. Fling 2. Leading edge moves away, air rushes in, increasing lift. Fling 3. New vortex forms at leading edge, trailing edge vortices cancel each other, perhaps helping flow to grow faster. Weisfo 1973. Governing equations. A wing moving in fluids experiences a fluid force, which follows the conventions found in aerodynamics. The force component normal to the direction of the flow relative to the wing is called lift L, and the force component in the opposite direction of the flow is drag D. At the Reynolds numbers considered here, an appropriate force unit is 1/2 rho U2s, where rho is the density of the fluid, s the wing area, and u the wing speed. The dimensionless forces are called lift CL and drag CD coefficients, that is, 8. CL and CD are constants only if the flow is steady. A special class of objects such as airfoils may reach a steady state when it slices through the fluid at a small angle of attack. In this case, the inviscid flow around an airfoil can be approximated by a potential flow satisfying the no-penetration boundary condition. The Kutta-Joukowski theorem of a 2D airfoil further assumes that the flow leaves the sharp trailing edge smoothly, and this determines the total circulation around an airfoil. The corresponding lift is given by Bernoulli's principle, Blasius theorem 8. The flows around birds and insects can be considered incompressible. The Mach number, or velocity relative to the speed of sound in air, is typically 1/300th and the wing frequency is about 10 to 103 Hz. Using the governing equation as the Navier-Stokes equation being subject to the no-slip boundary condition, the equation is 8. Where U, x, t is the flow field, p the pressure, rho the density of the fluid, nu the kinematic viscosity, ubd the velocity at the boundary, and s the velocity of the solid. By choosing a length scale, L, and velocity scale, U, the equation can be expressed in non-dimensional form containing the Reynolds number, ρ = Ul nu. There are two obvious differences between an insect wing and an airfoil. An insect wing is much smaller and it flaps. Using a dragonfly as an example, its chord C is about 1 cm, 0.39 in, its wing length L about 4 cm, 1.6 in, and its wing frequency F about 40 Hz. The tip speed U is about 1 meter per second, 3.3 feet per second, and the corresponding Reynolds number about